everybody. My name's Jeff McDermott. I'm a Science and Technology Advisor with 10x Genomics, and I just want to start by saying thank you so much for being here with us today. We really do appreciate the time that you've taken out of your day. I just have some really quick slides to showcase the 10x Genomics product portfolio, which can, of course, be utilized within the plant and broader agricultural research. The slide just contains some important information about forward-looking statements that I may be making today. So to make any progress in unraveling the complexities of biology, you're going to need the right tools at your disposal. And you need technologies that provide insights at that fundamental resolution of biology, which is, of course, the resolution of a single cell. And it's that need that underpins the direction we take when building our products at 10x Genomics. And as you can see, we built 3 platforms that each allow you to take a look at single cell biology with a unique perspective. So we have the Chromium single cell, which, as the name suggests, enables high throughput single cell analysis, allowing you to profile transcriptome-wide gene expression and other molecular features at single cell resolution for deeper insights into cellular diversity and function. Next, we have Visium Spatial Transcriptomics, and this provides spatial whole transcriptome gene expression mapping at single cell resolution, allowing you to explore tissue architecture and study gene activity within the precise spatial context at tissue section level. And then finally, we have Xenium In Situ, which delivers in situ high-plex RNA analysis directly within the tissue sample itself, enabling subcellular localization and quantification of hundreds of targets for advanced cellular profiling and spatial biology studies. This slide, the numbers on here are a little out of date. They were put together in September of 2024, but what you see here are plant-focused research papers that have utilized at least one of the 10x Genomics products that I just introduced. So as you can see here by this ever-growing body of work, all 3 of our 10x Genomics platforms can be used with plant material. Without diving too deep into the literature, I just want to showcase some common examples of why researchers have been utilizing the platforms from 10x Genomics in their plant studies. Shown in these next few slides are the main advantages of using single-cell and spatial transcriptomics in plant studies. I'll begin with studying cellular heterogeneity with precise resolution, and plants obviously have highly heterogeneous tissues where different cells within the same tissue or organ can behave differently depending on their location or developmental stage. So single-cell transcriptomics allows you to analyze the gene expression profile of individual cells, and by isolating and profiling individual cells, it becomes possible to identify rare or specialized cell types, such as those involved in defense mechanisms or nutrient uptake that would otherwise be masked in bulk RNA sequencing data. You discover rare and novel cell types, and single-cell transcriptomics has led to the discovery of new cell types and states within plants that were previously uncharacterized. These may include intermediate cell states during differentiation or rare cell types involved in specific physiological processes. And then spatial transcriptomics can be used to complement single cell findings by showing you where these novel cell types are located within any given one tissue. We're mapping gene expression in the tissue context. So speaking of spatial transcriptomics, this technique provides a 2D view of gene expression by maintaining the tissue architecture. It helps you as a scientist to understand how genes are expressed in specific spatial regions of plant organs, such as the leaf or root or stem, in relation to one another. This is important for studying how environmental signals like light and temperature and pathogens affect gene expression in different parts of the plant. Unraveling gene regulatory networks, and by analyzing gene expression at the single cell level, it becomes easier to construct these gene regulatory networks for specific cell types or regions within the tissue. These networks help you understand how different genes interact and control plant development and physiology, and this information is vital for identifying key regulators of important traits, such as disease, resistance, or growth rate and reproduction. We have understanding developmental processes, and plant development involves complex interactions between different cell types and layers within the plant tissue. So spatial transcriptomics allows for the mapping of gene expression patterns across the tissue whilst preserving their spatial arrangement. And it's really particularly useful for studying processes like root development, leaf differentiation, and floral organogenesis, where the position of a cell in relation to others is crucial for its function. Exploring plant environment and plant microbe interactions. Plants are highly responsive to environmental stimuli such as water availability, light intensity, and pathogens, as well as nutrient levels. So single cell and spatial transcriptomics allows you to understand how specific cell types or tissue regions respond to these external signals. For example, the response to drought stress in root cells can be profiled at much finer resolution, enabling identification of key regulatory genes and pathways involved in this stress tolerance. Gaining insights into plant reproduction, spatial and single-cell transcriptomics are particularly useful for studying reproductive organs, flowers and fruits and seeds, and that's because these tissues often contain highly specialized cells. And understanding the gene expression patterns in individual cells during key events like pollen development and fertilization and seed formation can provide insights into plant breeding and crop improvement strategies. And finally, improving crop traits with high throughput functional genomics. And both single cell and spatial transcriptomics can be scaled up to perform high throughput analyses of gene expression across multiple plant species, tissues, and conditions. And this provides a comprehensive view of plant gene function and its regulation across diverse biological contexts. So these technologies can be applied to crop species, enabling you to pinpoint the genes responsible for desirable traits, such as increased yield or stress resistance or even nutrient content. So hopefully it's clear just from those few snippets of data that single cell and spatial transcriptomics is transforming plant research by providing high resolution insights into the complexity of plant biology. These technologies allow you to explore the diversity of cell types, gene regulatory mechanisms, and environmental responses at unprecedented resolution, opening up new possibilities for understanding plant development, stress responses, and improving crop traits. So if you've been thinking about applying 10x Genomics single cell or spatial tools for your plant research, or you're truly inspired by what you see here today with our speakers, then certainly don't hesitate to reach out. We'd love to have a project discussion with you and see what's possible. So with that, again, I'll say thank you very much.
It was terrific. See you next time in one of our webinars.
Thank you.
Thank you.